Okay, thank you for the invitation. The title of my presentation is, are there any similarities between the effects of drugs of abuse and the development of syndrome E? Well, I think this may be an interesting question because, as we heard, drugs of abuse are able to have an extremely powerful possibility to modify personality. They facilitate violence, and it may be interesting to see how they work, and probably there may be some clues to understand how it can go to a sort of syndrome E in certain cases. Well, we don't know, we'll see. Well, the first thing, the chance we have as neuroscientists is that drugs of abuse act not on the 86 billions of neurons in the brain, but only on a minority of neurons which release dopamine, noradrenaline or norepinephrine, and 5-HT, that's serotonin, 5-hydroxytryptamine. So this neuron seems to be very important in all the psychic outputs of the brain. So how does it work? This is the first question. Well, they work at this level, at the synaptic level, the communication between the pre- and the post-synaptic neurons. So, for example, cocaine is here you see a terminal dopaminergic, for example, dopamine or noradrenaline, with the small circles are the neuromodulators. They are released in the synapse and they go to the postsynaptic receptors. Well, to regulate this density of neuromodulator in the synapse, you have a system which is called a reuptake. This dopamine is going back into the presynaptic neurons. And what cocaine is doing, for example, it blocks this reuptake phenomenon. And therefore, the density of dopamine or noradrenaline or 5-HT in the synapse is increased, and therefore you have an increased stimulation of the dopaminergic or noradrenergic or 5-HT receptors, and then the message is passing through. If now we go to methamphetamine or amphetamine, because we've been talking of Pervitin and methamphetamine, the system is almost the same. There is a small difference. Let me see. Small difference. It doesn't. It's this one. Some small difference between amphetamine is going inside the presynaptic neurons and chases the dopamine, noradrenaline from the vesicles, which protect the neuromodulators, and therefore there is also a blocking of the reuptake, but the density is increasing because there is a chase of this dopamine coming from the vesicles. So it's a slight difference with cocaine. Cocaine doesn't go into the neuron. Amphetamine is going into the neuron, and the stores are depleted very quickly. So you have to refill the vesicles after amphetamine. So if we go to the anatomy of the system, we see that it's very specific. You have in the mesencephalon, or is behind the bulb pond, you have 5-HT, noradrenaline, dopamine in red, which are going to the forebrain and in a very divergent fashion. And I think that's interesting to know these neurons are extremely, not only a minority, but extremely different from the other ones, the 86 billions of neurons. I tell you again that it's only 50,000 neurons which synthesize. And you have here 5-HT, noradrenaline, dopamine, much less than 1%. And here we have the neural networks with 99% of neurons which release GABA or glutamate. And this system is working like a mobile phone. You have an emitter and a receptor, and all that is extremely complex. You get networks, and these networks are modulated by these neurons, which are working like a radiophonic system. That is, you have an emitter which is in the mesencephalon, and anyone which has a receptor in the forebrain will receive the message. So you see, it's entirely different. And well, I'm going to try to illustrate this point because it's extremely important. Here you have a sort of schematic view of the brain with the spinal cord, thalamus, subcortical structures, limbic structure, septum, amygdala, hippocampus, and then the prefrontal cortex. And what we can do is try to put on this scheme, we can try to put the 86 billions of neurons. It is going to be very difficult. You're going to find my scheme is extremely complicated, but of course it's a gross simplification. You see what you can find, that's sensory neuron, auditive, visual, they got the thalamus, then the visual cortex, auditory cortex, and somatosensorial cortex. This is associated, all these information are associated, go to the prefrontal cortex or to the premotor or motor cortex. You can have output with motor neurons or eventually neurovegetative terminals with the hypothalamus. But in this scheme, we have no dopamine, we have no noradrenaline, we have no 5-HT. And this is the most important part of the brain, of course. But how are these modulators? How are they distributed? And you will see that it's entirely different. You see that dopamine is red, noradrenaline is white, and 5-HT is in yellow. And you see that cell bodies are located in the mesocephalon, and they don't pass through the thalamus, it's not a problem. They go in very divergent way to the forebrain structure, subcortical or cortical structures. So it's a sort of secret which is superimposed to the previous one. And you could say, when you see that S is, wah! You could say that they are all identical. But when I have shouted now, you have increased the activity of your noradrenergic neuron, precisely your noradrenergic neurons. You see, you have a locus ceruleus activation, and you have modified the signal-to-noise ratio in your brain. This noradrenaline is supposed to control the input from the outside, whereas you have also activated the 5-HT neurons to protect the nervous central system from this activation, brutal activation. So these 5-HT neurons are protecting the system from outputs. And then dopamine 1 are integrating all the system. So this can be seen here. This noradrenaline system select and amplified external information. There might be a danger when I have shouted. So there is few milliseconds, 30 milliseconds exactly, between the shouting and the jump you have done, all of you, most of you anyway. And 5-HT neurons maintain and protect the central nervous system, while dopaminergic neurons decode the significance of the external and internal information. And if I say all that as just to summarize what we think about this regulation, modulatory regulation of this dopamine, noradrenaline, and 5-HT. You have here a sensory afferent, thalamus, then you go to the different cortical area, sensory area, while noradrenaline and 5-HT are simultaneously activated. They give a significance to the signal coming from afferent for perception, external perception. They are going from the caudal part to the prefrontal part, the cortex, and then you have activation of dopamine, which is actually giving a sort of hierarchy between cortical and subcortical structures in order to get an output which is related to the significance of the afferents, sensorial afferents. The significance has appeared in the cortex from the caudal to the prefrontal part. So you understand that in this way, dopamine might be very important because it's the last point, it's the last modulatory before the output. And indeed, most people, most scientists consider that dopamine is extremely important for different reasons. The first one is, I told you before, well, quickly, is that all drugs of abuse release dopamine in the central nervous system. This is an importance. The amplitude of the release depends on the drugs, but all of them release dopamine. And the second point is that when Olds and Milner, two American scientists, have descended electrode in the rat brain and given the possibility to the rat to stimulate some parts of their brain by pushing on the pedals, they found three results. The first one, animals had a sort of aversive reaction to the stimulation, the electrical stimulation. Second groups had no effect, not at all. And the third one got to the pedal and went on going to the pedal, having more and more electrical stimulation. And the animals could not stop from activating this pedal. And if you leave them with the electrical activity, they die. They die not because of the current, of the electricity, but because they don't go anymore to eat or to take any water or anything. So they die because they have been an activation which makes them, would say, happy, pleasure, satisfaction, anything, but they are okay, no problem, and they die because they go and push this pedal. So that's interesting to see where were these electrodes which induced this activation. And the first one was in the hypothalamus, which is not a surprise because you have sort of sexuality and food and thirst and all, which is regulated by the hypothalamus. But the second point was a surprise because at that time nothing was known about dopamine. And the second point was here, which is called the ventral tegmental area. And it is a point where dopaminergic cell bodies lie and send their axon in the forebrain in a very divergent way, as I've shown you before. So this point seems extremely interesting. The animal, when it stimulates this point, gets a reward, and this reward is so strong that they don't need anymore to go and eat. So they die from that. The only way to stop them from dying is to stop the electrical current. So this is an important point. And the last point which made us very interested by dopamine is a fantastic work of Wolfram Schultz, who took a monkey and recorded in the ventral tegmental area where dopaminergic cell bodies lie, recorded the activity of the cell of these neurons in a monkey, awake monkey. And the monkey had a sort of plastic tube which was going under his tongue, and sometimes there was just a drop of apple juice. And when the monkey thinks apple juice is fantastic, he likes enormously, huge preference for apple juice. So you see the activity of dopaminergic neurons before the reward. You get the reward here, apple juice, and then you activation of the ventral tegmental area. Now, if 1 second before the reward you light on a red light, then you see that after some trials, the animals activate the dopaminergic neurons when the light is appearing and not when the reward is there. This means that dopaminergic neurons are able to anticipate the reward. And then the last part, the reward is predicted, but it doesn't send the apple juice, apple drop. And then you see there's still the activation, but few milliseconds after the time where the reward should have arrived, there is a decrease of dopaminergic activity, which indicates that the animal has had frustration. Altogether, it seems there is a sort of reward circuit in the central nervous system, which can be summarized this way. You have a certain number of cerebral structures, prefrontal cortex. We could say orbitofrontal cortex, as Rolf indicated this morning, septum, nucleus accumbens, hippocampus, amygdala. All these structures are interconnected, and they send information to the hypothalamus, which by itself goes to the ventral tegmental area, which contains dopaminergic systems, and goes up. Dopamine is divergently going to the reward circuit. It's interesting to know that hypothalamus doesn't receive any clear information which has not been filtered by these four structures. So if you give the possibility to the animals to stimulate VTA, ventral tegmental area, activating dopamine, then you get a reward. Then the animal considers that everything is okay. Okay, but if you give drugs of abuse and you release dopamine, then you have the same type of phenomenon. You get reward and everything is okay, even if on the side there is problem. If you have problems, you take drugs, all the problems are going away. So that has been done on animals. And the question is, what would happen in man? And the question tried to be studied by a Dutch team, which tried to imagine which could be the best pleasure you can imagine for a man. And he found that orgasm could be the best thing to do. So they have done image analysis of what's happening in the brain during orgasms. And you see that what they see is that there is a strong activation of the mesencephalic diencephalic zone, which is the ventral tegmental area, VTA. So it's okay. It looks like what is happening with drugs or with autostimulation during orgasms. And then they went a little further and looked at the brain, and they found most orbitofrontal cortex probably, but they found that temporal cortex was activated and frontal cortex was activated. Only the right, which is interesting, not the left. I don't know why, probably because they were right-handed, I don't know. Well, anyway, well, it should be left-handed, but it doesn't matter. And the point is that everything is okay, but they found something extremely interesting, which may be interesting for us today, is that when they looked at the different structure, they found that one structure was deactivated. And the structure which was deactivated is the amygdala and also the entorhinal cortex. And this is interesting because it seems that to get the pleasure, that the pleasure be able to feel it, you need an activation of ventral tegmental area, seemingly, but dopamine, probably, but also prefrontal cortex, orbitofrontal cortex, and also you need to deactivate amygdala, or all of you probably know that amygdala is coding for fear, for risks, for danger, and for anything which is aversive. There is a risk, and if there is a risk, and if your amygdala is activated, you can't have pleasure. So if you have all the three structures activated, VTA, prefrontal cortex, and amygdala, the system is blocked. But if you can deactivate amygdala for any reason, then you can have pleasure. The theory is that the prefrontal cortex and the orbitofrontal cortex probably is activated in such a manner that it decreases the activity of the amygdala if everything is okay. But that may not be the thing all the time. So it means that to get the pleasure, you must have this sort of disequilibrium. And now, this is the most important point I want to emphasize. This is the way scientists use to say that something is rewarding, a drug of abuse is rewarding or not. This is what's called a place preference, conditioned place preference. You see two compartments. One is slightly different from the other, and you can put an animal, a rat, for example, and give it cocaine in this place. Then the day after, you give him physiological serum. And then the day after, and that you give him cocaine here, and then the day after, you give him physiological serum, and you do that for 4 days. Then on the 5th day, you leave the animal in this place, and you wait. And you measure the time the animal is going in this place, and this animal is going in this place. If the drugs that you have injected to the animals, for example, cocaine, is rewarding, it will stay in this place. If it is not, or if it is aversive, it will go here, or maybe 50-50. Actually, all drugs of abuse induce a preference for this side if it has been injected in this side. And that is the most important point. When they have done this experiment with a rat or mice, actually it was mice, and on the first day, you put a mice in here, and then another mice here, but a smaller one. Then the big mice attacked and strangled the small one. They leave them for 10 seconds. They don't want to die. And then the day after, the big mouse is put alone in the other way. The day after, 2 mice are put here, struggled again, and so on. And when they put the mouse after 4 days here, the animal is preferring this place, which means that the struggle is a reward. The struggle is rewarding for these animals. So it's clear there is something which puts the animal to go to struggle. It likes it. It likes it. And this is even worse, I would say, even worse. There is another way to measure the rewarding effects of drugs of abuse. It is called the self-administration. The animal learns that if it puts its nose in the active hole 5 times, 5 successive times, then the computer records the things and it gets an injection of cocaine. The animal is learning this task very quickly and very easily. On the about fifth day, he is able to do the thing, and then he will get to the inactive hole, but 5 times in the active hole. And the experiment, which has been done by another team, was at the 5th time, they give the possibility to open the cage. The cage is open, and another rat is coming inside. And they struggle again. And you find very quickly that the animal learns that when it goes 5 successive times in the active hole, he gets another one to struggle with it. So there again, it means that the animal is extremely rewarded by struggle, by violence and struggle. Moreover, if you give pharmacological drugs which block the rewarding system, then the animal is not doing anymore the task. What does that mean? It means that depending on the possibility of this equilibrium between prefrontal cortex, ventral tegment area, and amygdala, there can be, in some cases, the possibility to be rewarded by violence, by crime, by struggle. So the problem is, how is it possible to block the deactivation of amygdala in certain conditions? And this probably is a question that I can ask to everybody. But the thing is, if individuals are trained or conditioned to block this deactivation, to block the activation from the cortex to the amygdala, then they will not have any pleasure. But if the 2 systems are working, that is, if by crime you can activate prefrontal cortex and deactivate amygdala, that is, you are not anymore feared by what you are doing, you are not risk of danger, in this condition, then you get rewarded, and it works as if you were taking drugs of abuse. So you get exactly the same sensation, feeling, that if you were getting drugs of abuse. So how can it be possible to obtain this result? Normal people do activate their amygdala when they try to perform crimes, so they don't get pleasure. But some people are able to deactivate their amygdala even though on the beginning they were not. And this deactivation may occur extremely quickly and depends on trained or conditioned situation, or maybe uncertainty. We heard about uncertainty or stress or different possibility which may induce that the excitation induced by crime and violence, which occurs. I think there is excitation. Everybody is excited by crime and violence. The brain is arousal, or in hyper arousal if you have to kill someone. But you don't like it if you don't deactivate your amygdala. Therefore, the crime and violence may act as a drug of abuse, induce euphoria, pleasure, and if you repeat the thing, you may become addicted to crime and violence. And now, to finish this presentation, I'd just like to go back to syndrome E and see that, indeed, we can understand very easily the different definition of syndrome E by Dr. Fried. You have a repetitive acts of violence, obsessive ideation, perseveration, diminished affective reactivity. We are exactly in here. If you are able to inhibit your amygdala in this condition, compartmentalization, group contagion, rapid habituation, and environmental dependency. And most importantly, there is no problem about your working mind. You have still all your capacities because your cognitive functions are still absolutely intact. Language, memory, problem-solving skills. There is no reason that because of the capacity to inhibit amygdala would change your problem-solving skill. Well, I try to find the hypothesis. Thank you.